Excited to be here. So, hello everyone, I'm Sarah. As Mr. Ripley said, I'm a graduate fellow at the University of Alabama, and I'm so excited to be joining you from here today. So please confirm that you can hear me properly and you can see my screen.
Yes, we can. I can.
We can hear you, please.
I can hear you.
Okay.
Yes, I can hear you.
Can hear you.
All right. Okay, so welcome to this 5-day workshop on bioinformatics. So biology is rapidly growing to accommodate data science as a subfield, and we're really generating billions of biological data points from genes, proteins, and other molecular analysis that can be combined into large files and systematically studied. So analyzing this data will give us more knowledge and understanding about living organisms, including human health, biotechnology, crops, and livestock, depending on where you want to apply it. So this course will exactly give you an overview of bioinformatics for students, the biologists, and clinicians. So the influx of data and the different associated methodologies and data-driven research makes bioinformatics for life sciences as prominent as the emergence of computers for the tech industry. You can see here that this bioinformatics is more like an interdisciplinary field. I like to call it that because it involves applying informatics, statistical tools, and like analyzing biological data, getting biological information. This is not to say that people in chemistry don't use it or other fields, but mostly it's all about like getting information about particular living organisms, and biology is more about studying living organisms. So bioinformatics is a convergence of biology and data analysis, where both are equally important, as we'll see in the next few slides. So it's always a big question in bioinformatics about which do I have to be really good at? Is it the biology part? Is it the computer science part or the mathematics part? So we'll see in the next few slides that every part is equally important. So we could have said probably just statisticians or computer scientists or engineers need bioinformatics. But the truth is that more biologists and clinicians are actually expected to understand their own data, handle this data, and publish and succeed in basic and translational research. So we can take data from biology-related fields and use this to make informative decisions. So for example, in plant biotechnology, we can decide to study polyploidy, that is like the duplication of genomes and all that, and analyze these plant genomes using informatics tools. So this can give us a whole lot of information on where and where this duplication has occurred, how these polyploid events have resulted in plants acquiring resistance to some environmental effects. So in evolutionary biology, we really use this a lot, and you can just know how genes have evolved over time. Okay, so bioinformatics is the intersection of informatics or the science of processing data for storage, retrieval, and then the biology of living organisms. So informatics itself is the science of processing data for storage and retrieval. So while biology is the study of living organisms. So bioinformatics therefore can be said to be a sub-discipline of biology and computer science. And it's concerned with the acquisition, storage, analysis, and dissemination of biological data. So both are equally important, like I said. So, but it can be quite tricky to like know where to actually get those skills from and where to actually concentrate, because it's just like you merging two different disciplines.
Hello, Sarah, please, can you be a bit more audible?
All right, I was about to say that too, because the voice kind of getting faint, by the way. Yeah, but
is it a network issue or is just me not talking loud?
Now it's clearer. Okay. I think you just need to just speak a little bit louder.
All right.
Yeah, she was speaking like with her nose. I wasn't hearing her very well. So try to be audible, please, so we can hear you.
Okay.
Thank you.
So so this field has also developed tremendously in the last several decades, transforming the life sciences and enabling biotechnological tools and empowering biomedical research. So as a result, we have many new discoveries that include new treatments, diagnostic assays, design strategies for experimentation. I hope it's better now.
Yes,
Can you hear me clearly?
yes, more audible. Yeah, thank you.
So, okay. So importantly, today there is a growing appreciation among biologists, biomedical scientists, and clinicians and researchers for data. So this includes new types of data generation tools, computational methods of data organization and analysis, and also visualizing this data that we are generating. So this data, analyzing this data can show you statistical patterns in the context of a biological phenomenon. So you can see a whole lot of things that we do here with this picture representations, the distribution, the map, box plots, and statistical tools that is used in telling a story about your data. So this science is based on high throughput technologies that generate billions of data points on genes, proteins, and molecules that characterize biological systems. So we compile this data into repositories that can be systematically studied to interrogate and establish trends for so many applications. So the biologists carry out sequencing to extract this data from genetic materials. Then we assess living organisms at different levels. So first we have the phenomics. So this, the first level that researchers actually assess living organisms is this level. So it's defined as the changes that are seen in an organism resulting in variations in the phenotype during the lifespan of the organism. So it's more about the physical characteristics that you can see in a particular individual. So the set of phenotypes on a wide scale dealing with the organism's physical and biochemical traits. That's when we come to like studying the phenomics of a particular organism. Then we move to another level, which is the genomics level. So at this level, a genome is like the complete set of genetic information of an organism. And genomics is about studying the whole genome of these organisms and utilizes a combination of DNA sequencing methods and bioinformatics to sequence, assemble, and analyze structure and function of the genome. If you're not quite familiar with sequencing, we will get into that in a minute. So next we move to epigenetics. So epigenetics, on the other hand, is the set of changes in an organism that is brought about by modification of gene expression rather than alteration of the genetic code in an individual's DNA. So epigenetics is about DNA methylation or modification of the DNA that results in different expression, but it doesn't mean we're changing the genetic code. Then we move to the transcriptomics level. At this level, we're studying the RNA gene expression. So it's a total set of mRNA transcripts that is being obtained from transcription of genetic information. So this determines the gene expression in a set of tissues at the respective time and condition. So for transcriptomics, it's more about gene expression at a particular condition. So when researchers want to do with transcriptomics data, they are really talking about what condition, what specific time they took that data. So the comprehensive analysis of this complete RNA transcript for a particular cell, tissue, or all organisms at a particular time under a specific condition is what we call transcriptomics. So these different levels are the different ways researchers can analyze living organisms and assess these data points. So we could be having genomic data, we could be having epigenetic data, that's DNA methylation data, we could be having transcriptomic data, and what have you, just to use bioinformatics tools on them. Okay, so to the sequencing aspect, we have the Sanger sequencing, also known as the chain termination method. And the sequencing is all about like determining the nucleotide sequencing, the sequence of the DNA. Yeah. So Sanger sequencing is like one of the oldest methods, and here the DNA sequence of interest is used as a template for a special type of PCR that we call the chain termination PCR. So this works just like the standard PCR, but the major difference is that you're adding modified nucleotide. You're adding a modified, they call them dideoxyribonucleotides, DDNTPs. Yes, that's what you're adding to the whole system. So in the extension step of chain termination PCR, the user sort of mixes a low ratio of chain terminating nucleotides with the normal nucleotides that you have in PCR reaction. So the difference between the normal nucleotides and the deoxyribonucleotide is that the DDNTP lacks the 3-hydroxy group, and that's needed for phosphodiester bond formation. So when the DNA polymerase in the reaction incorporates the DDNTPs, the reaction ceases and we don't have the extension stage of the PCR. So the result of chain termination PCR is like millions to billions of oligonucleotides of the DNA sequence of interest that is terminated at different lengths. So in the second step, as you can see in this flow chart, is the chain terminated oligonucleotides are separated by sizes via gel electrophoresis. So the last step simply involves reading the gel to determine the sequence of the input DNA based on how they've been separated on gel electrophoresis. You can determine what those sequences are. So you can just visualize the DNA bands by autoradiography or UV light, and the DNA sequence can be directly read of the X-ray film or gel image. So, however, standard sequencing is best suited for medium to low throughput targeted sequencing. So for higher throughput DNA sequencing, we use different methods that have emerged in the last decade, and they're known as next generation sequencing. So next slide, we're looking at next generation sequencing. So the advent of high throughput sequencing technologies has not only created a whole lot of huge data set, but has also paved the way for a new era in genomics and its application in various fields. So as I've mentioned earlier, we are generating billions of biological data points on gene, proteins, and molecules. So from this next generation sequencing machine to generate a whole lot of biological data, and they are being published from laboratories, by researchers, research institutes, on various databases and procedures. And then we also have transcriptomic sequencing. If you remember, I mentioned transcriptomics, like RNA sequencing. So transcriptome sequencing is a major advance in the study of gene expression because it allows you to take a snapshot of the whole transcriptome rather than a predetermined subset of genes. So at a particular point that the gene is being expressed, you can take a whole snapshot of it and analyze that data. So whole transcriptome sequencing provides a comprehensive view of a cellular transcriptional profile at a given biological moment and greatly enhances the power of RNA discovery methods. So to study this data, we'll learn about processing pipelines that turn raw reads into structured data ready for analysis. So after getting your data from the machine, from the sequencing machine, you really need to structure your data so you can load it into softwares, into environments where you can analyze your data quickly. So let's really understand the basis of like NGS sequencing, next generation sequencing. So the whole genomic DNA is isolated from the tissue, and then it is sort of shared into smaller pieces, then placed in a flow cell. So inside this flow cell, cDNA fragments are amplified using bridge PCR amplification. This is a different PCR amplification method rather than chain termination method that we use in standard sequencing. So in this bridge PCR methods, you can have the strands of the DNA being attached to the flow cell, and then they are inserted into the sequencer. So the sequencer uses image analysis to capture each letter in the flow cell and sort of read what exactly the letter can be. So through visual patterns that different of those bases have, A, T, G, C, we can always, the machine can always record this and say, okay, this is A, this is T, this is G, and this is C. And then at the end of the whole process, you have your data in this format. Like you have the whole lot of 80s that you can analyze. So eventually to produce reads that typically from 30 to 300 base pairs. So how do we really get this data? How do we get in a format that we can analyze? So these are the different formats that we have for next generation sequencing. So for example, the FASTQ files provide us reads from the sequencer. It's a text-based file format that stores nucleotide sequencing. But it stores the reads with the quality score. So quality score is more like the confidence we have in the machine saying this is A, this is a T, and this is a G or C. So for the fastest format is a text-based format also for representing DNA and protein sequences, but typically it's always the reference genome. So the reference genome is usually in the faster format, and when we are sequencing non-reference genomes, mostly it's the faster format. Then we have the genome transfer format, which contains genome annotation. For example, the GTF will store the start and end of each genomic element, and then we can get exon information, isoform, and gene information from the GTF file. So in our analysis, what is the reference genome, the new whatever genome we're currently sequencing, and depending on what you're doing, we might need the genome annotation file. Then we also have the SAM and BAM files. So the SAM file, that's short for sequence alignment map. It represents the alignment of these roots on the reference genome. So it usually starts with the header section, followed by alignment information as tab separated lines for each read. So you can always look at the SAM or BAM files and read it. Yeah, there are ways to read it. It has different codes and reading the different sections of the genome. And then the BAM file is just something like the SAM file, but it's just in binary format. So depending on what the researcher is doing, they use the different formats of the files from the sequencing machine. So that's what we actually use computational tools to analyze. So at this point, I would like to take questions and just possibly, and also check the chat section. If I'm missing anything in the chat box, or they're just no questions.
Well, basically in the chat box, we just have comments that are saying that your connection is very clear and you're very loud, and that's the most of what I am experiencing. But yeah. So
okay, someone raised a hand.
Okay.
Do they want to ask questions or do they
Right, yeah.
Chat box?
Please, you can unmute yourself, Olu Benson. Your hand is raised.
Okay, am I audible now?
Yeah, you are.
Okay, so my question just goes back to the stages of the classification of the omics, phenomics, genomics, epigenomics, transcriptomics. I was asking about proteomics. Is there anything like that? And why is it not part of the list also?
Oh, proteomics is more like protein, like getting protein sequencing, protein structures and all of that. It's actually part of it. We can always put our proteomics in probably the studying some of the phenomics and all that. We also have metabolomics, right? So
yeah.
This is like 4 different levels. So most times they're separately on their own. We have the proteomics and the metabolomics. So most times you just say they are genomics because the protein is maybe like the protein information from the genome. So most people just say omics for the general term, and then you can have genomics, proteomics, metabolomics, transcriptomics, depending on what you are doing. So it just depends on how different researchers actually classify things. Yeah.
Okay, that's fine. Thanks so much for the answer.
Yes.
Before I call on the TechnoSpark, I don't know your name. There's a quick question here that's asking, okay, one of them is asking for the difference between the faster and the FASTQ. Then another is asking about the difference between BLAST and FASTA for inquiry. So, okay.
Okay, for FASTA is different from FASTQ. FASTA just gives you the identifier and information about the sequencing. But for FASTQ format, you have the sequence and you have the quality score. The quality score is usually important to researchers when we do bioinformatics to like know how confident, what's the error rate, how confident is our analysis before we start anything. Because once there's an issue with your sequencing method or the sequencing data, whatever thing you do is going to be affected. So it is good to really have the quality score. So that's the difference between the FASTA and FASTA. Is that fine?
Yeah, thank you so much for that. Let me call on the TechnoSpark. Please next time, just name yourself. Are you there? Can you unmute? Yeah.
Okay. Good afternoon. Sorry for that.
Yeah, what's the name?
Okay, Chibuka is my name actually.
Okay, yeah. Continue.
So I came a little bit late, but I like the presenter to shed more light on the FASTQ file. Thank you.
Okay, that's fine. Like I said, like I just mentioned now, the FASTQ file gives you the quality score of your reads. So we have different sequencing technologies for the We have Sanger sequencing, we have Illumina, we have PacBio, and all of that. So they have different read and yield rates, and they have different error rates. So I think PacBio is a long read sequencing technology, and it has, I think, a higher error rate. So this quality score helps you to like check the quality of the data you're getting. You don't just, it's really, before you start a bioinformatics analysis, the quality of your data is really important. We're going to check that soon, like the value of your data, what value can you get from this data? So FASTQ files sort of gives you the quality score of the data you're working on. So each, for example, we don't have a bigger picture here, but if you, for example, we have an A, under it, we have a particular computer encryption that tells you the quality of that A that is given. If it gives you a T, it gives you the quality of that T. Is that fine?
Yeah, thank you. Thank you.
All right. Before I call on the next person, I also believe that in your FASTQ file, you can find other metadata that may be lost already in your FASTA file. So just take note of that. So I would call on Silas. Silas, can you unmute and question?
Okay, thank you, Sarah. Thank you for the beautiful presentation. I really enjoyed your presentation. I would like you to throw more light on the Sanger method of sequencing and the next generation sequencing. Are there types of next generation sequencing, or just a generalized kind of sequencing? Please, you can throw more light on that one. Thank you.
Okay, yeah, that's fine. So the Sanger sequencing is like an older method. Some labs still use the Sanger sequencing. So it's more of an old method that is not really efficient. It can't give you a high yield. And currently we have billions of data points, and we need something that is quite fast that can be used for high throughput data. So we have the next generation sequencing is like the new generation of sequencing machines that we have. So we have the Illumina short read sequencing, the PacBio, Nanopore, and all of that. So those are like the recent ones that people really use. Illumina is mostly for like short read sequencing, and then we have PacBio for long read sequencing. And so labs still use the Sanger sequencing. So the difference is just that they give you different error rates, they give you different yields of how many data points they can actually give you at a particular time, how fast they have to give you the data. And some are more expensive than the other.
Well, thank you so much for that answer, Sarah. I would just love to add that maybe you skipped a part of the question that Mr. Silas is actually asking.
Okay.
I believe that he was trying to know to what extent, to what what category, what limit does the next generation sequencing have? Because since the next generation sequencing actually surpasses the Sanger sequencing, I believe that depending on the area that the researcher wishes to actually continue, to research, your next generation sequencing application, I want you to take note of the fact that I just said application, can actually go through, can actually help you to explore whole genome sequencing, whole exome sequencing, targeted sequencing for specific studies. It can help you to go into epigenomics. It can help you to go into RNA sequencing of transcriptomes. In case you are that kind of person that's interested in CNVs and SNVs, it can take you into single nucleotide. It will help you to go deeper and deeper. Yeah, and the PCR for NGS, you can go even into details that relates to your proteomics, the details that relates to your metabolomics, and a lot of details can actually be unlocked based on the different applications of NGS applications. So we just love to just add the fact that all these different things I've just mentioned are actually different specialization fields. If you are a botanist, for example, or you are a medical scientist, or you are someone that works with drugs and co, you will actually have to apply these methods differently. So at that point, let me just stop so that it doesn't look as if I've said a lot. So let me call on Paul. I hope I've answered your question, Mr. Silas. All right, let me call on Paul. Seems like Mr. Silas is not there. Mr. Paul, are you there? Your hands are raised.
Yes, sir, I'm there. Can you hear me, sir?
Yeah, I can hear you.
Okay. My question is, thank you very much, Sarah, for the presentation. My question is, which file format is best for next generation? You can see FASTA, FASTQ, GTF, SAM and BAM. So which one will be much more okay to work with?
Is best, okay.
It depends on what you're doing. So the FASTQ, like I said, is the most likely we're mostly using for the reference genome, the FASTA file for what you're doing. So most times you use the FASTA file. Then the GTF is the annotation file. So annotation is different from your sequencing rate. Annotation is just telling you like this particular read, what it is aligning to, and all of that. Then we have the alignment file that like gives the position of the genome and all of that. So it depends on what you're actually doing. When you sequence the data, you want to align it to your reference genome, you want to annotate the genome. So that's why we have different files. So once you finish your sequencing, most times you have your FASTA file. Or if it's a reference genome, most times you have your FASTA file.
Okay, thank you very much, Simon. Thank you very much.
Yeah, there are some other questions in the chat. I would love to ask that some of these questions are sent to WhatsApp group such that we can all touch the questions there. We can all see the answers there, and we will be able to answer them. One of the quick question I can see here is that you mentioned something like laboratory preparation. This person is interested in knowing what that actually entails. Please, over to you.
Like lab preparation before sequencing?
Yeah, kind of.
Yeah, so you have to like
library preparation.
Sorry, library
preparation. Yeah, so when you have your sample that you want to work on, you need to like create a library of your data. So you just have your DNA and you have the templates and the primer that you need, the adapter and all of that. So creating a library is more like creating your template and what you want to add nucleotides to, what exactly you are emulating and all of that. So you need to prepare your library before it's inserted into the flow cell, and then you have your sequencing process. Ayanfeoluwa, would you like to add to that?
Yeah, sorry, I was speaking and I was muted. I was just going to say that library preparation actually it just involves generating some collection of data fragment for sequencing. Let me say that again. It involves you, it requires that you generate a collection of data fragment, of DNA fragment for sequencing. So you actually need to know what you want to do. You don't just get random DNA fragment for sequencing. You should already have studied, have gotten to understand that, okay, this is the cluster of DNA fragment that I need to replicate, that I need need to pursue that actually have a potential to really solve some of these my questions. And the NGS library are typically prepared by fragmenting of dynamic data or cDNA samples, like getting specified adapters, like she mentioned, for both fragment ends. And it goes on. It also differs. Library preparation actually differs from the type of the NGS technology that you're actually using. If you're someone using the Illumina, the PacBio and Co, you will have to prepare it a little differently, and depending on your target too. If you are actually going to go in a direction of, let's say, the old genome sequencing, you will have to prepare it differently, and your number of base pairs are also going to differ. So I'm just going to stop there. Can I see other hands? Okay, I think I can still see Mr. Silas' hand. Are you there? Okay. I don't think, okay, I think he's muted already. I don't know what's going on there. Okay, so
sorry, it was an error, sorry.
Okay, it's all right. Let me just take this last question here. It's from Stella, and she was asking about the difference between, please explain SAM and BAM. That's going to be the last question, Sarah.
Yeah, so the difference between SAM and BAM is that one is in a binary format and one is not in a binary format. One is more in a text-like format, also, I would say. So the SAM and BAM file, like I said, the alignment, if they give you alignment information, so you can, I wish there was a bigger picture, you can really see maybe the flags, the end, where this genome is starting, where this position is ending. So we really use the SAM and BAM files in aligning whatever sequences to our reference genome. So you can use either of the two. I think I've used the SAM file and the BAM file, and it's just, they do the same thing. It just depends on what software you're using or what you want to use the data for, and which is just better for you to analyze. So at some point, I've found the SAM file to be better, and at some point I just aligned and used like the BAM file. So it just differs. They're just the same thing. They're alignment files, but they give you like different.
Thank you so much. Someone just wrote, these terms are new to me. So it's so interesting that you're actually meeting some new terms here. I'm so excited that you're actually getting this. I want you to write your questions now in the chat. Our campus ambassadors and our teams are going to take questions and paste it on the WhatsApp group so that we can answer most or all of your questions on the WhatsApp group. So Sarah, I would love you to continue with the slide. Continue, cheers.
Okay, yeah, thank you.
So before you move on, please. So I'm happy to see some of those comments from newbies and people commenting about not having good grasp of some of these terms yet. That is the purpose of this opening live session, and I can assure you by the time you start taking the courses, the asynchronous courses from tomorrow, introduction to bioinformatics, bytes and molecules, that will take us from the basis, what's the DNA, nucleotides, and all of that. The various file formats will also be discussed during those courses. So please let us wait for that, and let's be inspired and motivated with these, with the things we learn, the new terminologies. Let's take note of them against tomorrow. Over to you now, Sarah. You can continue so that we get to the end.
Thank you so much, Mr. Lippman. Thank you, Ayanfeoluwa, for the help with the session. So I'm going to run through the rest of the presentation, and I hope you find it interesting. Okay, so what makes data into big data? So over the past 20 years, there's been a phenomenal growth in the adoption of high throughput technologies like next generation sequencing and research, biotechnology, and clinical practice. And this has created exponential growth in publicly available data. So we need to understand why we call this big data. Why do we really need to start studying this? Why is the data big anyways? So there are 5 V's that's going to guide us in understanding why we refer to this data as big data. So the first is volume. The amount of data is huge. For example, yields information about tens of thousands of genes or millions of single nucleotide mutation variants. So it's really a lot of data to like go through. And we need like computer languages or computer techniques to be able to go through them fast and get all the information that we need. And then we have variety. So the data is unstructured and can be assembled in many different ways. So oftentimes the same phenomenon can be described in many different ways, depending on the type of sequencer you use, the preparation technique, or the lab setting of the lab preparation or the kind of lab that produced data. So we just have different, it's just in different ways. People do it differently. Some people use Sanger sequencing, some people use this sequencing method, depending on whatever they're doing. So then we also have a value. Like I said, what's the quality of this data? What value can we get from it? So the information we get out of the data is dependent on many aspects, including how much noise is in the data, do we have error rates in the technique, how complete is the information that we're getting from single samples. Then next V, we have veracity. Some samples can have low coverage, others high coverage, but not full sequences. So, and others might be generated by an older machine that has an high error rate. So, including all these data together, you have to include so many data to get a complete picture of what you're doing. So, this is why we also have big data. So, then velocity is the last part. So, and it's an important aspect because Sanger, for example, Sanger sequencing was replaced by shotgun sequencing. So, and eventually not single cell sequencing. So this field is evolving and you need to be evolving as it is evolving. You need new skills. As the skills are changing, they are also like changing, getting more into the new skills that we have. So we are always learning new things and have to adapt to this idea that new samples expand our understanding before we even have the chance to study everything from the data we have. So let's look at the history of genomics data a bit. So it's been 70 years since the structure and function of DNA was discovered, and 50 years have been spent sequencing the human genome. So in 2015, the 1000 Genome Project brought a significant number of whole genome sequence to the research community. So we have the sequence that many researchers can analyze. And then 100,000 whole genomes were sequenced by 2018. So today there's an ever-increasing number of data sets available to help researchers understand cellular processes. So most times bioinformaticians go to databases and get this data, they analyze them, get more information about biological data. So for example, recently in April, scientists from the Telomere Consortium, they reported a completion of the remaining 8% of the human genome. So it was just recently completed. The human genome, like sequencing the whole human genome, was just recently completed. And we also have researchers going into the Earth Biogenome Project, where they want to sequence every eukaryotic organism that we have on Earth, looking at about 8 million eukaryotic species in 10 years. So you can imagine if we have something like that, the amount of huge data that we have, and the need to like develop computational techniques to actually analyze this data and get reasonable information from it. And that's about research. You are learning every day. We really want to understand why things are this way. So when we get the data, we can analyze and get useful information. So once again, this program was designed to highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, and machine learning. So this processes are really transforming personalization of treatment selection and innovative algorithms that allows unprecedented capabilities for drug discovery, design of large molecules, and drug repurposing. So, but in the pharmaceutical industry, with chemists, pharmacists, and all of that, they are really going into analysis of big data because they're using in drug discovery in different parts of things that just deal with you improving living organisms. You have bioinformatics there. So it's not just for biologists, in case you do major in biology. So This program will give a comprehensive overview of bioinformatics for students, biologists, clinicians, and what have you. So the resources and the data we have, they tell us a very clear story that as a scientist, as a researcher, we are capable of like doing our own research and generating data and getting information. So now let's look at where you can find various types of big data and associated studies. So if you look at NCBI, we have a whole lot of publications on NCBI, PubMed, that different researchers upload their data to. So here you can search for almost all research publications in a particular field. For example, if you use the term liver cancer to get a search, you can see how it has grown like exponentially. For example, you use liver cancer. Publications of liver cancer really go exponentially in the database. So all biologists submit their experimental data in the different public repositories, and as a result of lots of data from experiment, as a result, we have a whole lot of data that is being generated that's been submitted to these repositories. So we have the GEO apart from NCBI, we have gene expression images, and I think GEO contains about 80,000 public data sets that's spanning different types of tissues, different types of organisms. So this data set are continuously growing in an exponential manner, as I said earlier. If you take the field of oncology, for example, there's a really fast growth, high throughput sequencing of cancer genomes and transcriptomes. So this has created a big data problem for cancer biologists and oncologists. So this will help them to understand the process of malignant processes, relationship between tumor genomic profiles and treatment response. So it's important to learn about these data types and how to efficiently process and analyze them. So by looking at this the backlog that I showed you here, you can see how many publications are there per year, and you notice the amount of work that's been done in recent years. Yeah, so there has been a remarkable shift over the last decade in the cost of generating molecular measurements, most notably DNA sequencing. There has been a really a different way that people generate this data and how much they actually generate it. So it has also affected transcriptomic data, proteomic data, metabolomic data, and all of that. So and a field that is really impacted by this generation of data is genomic medicine, where it is possible to move from generating reference or population of data to producing data from individuals. So concurrently, there have also been advances in the development of new algorithms and tools to integrate and interpret this data. So bioinformatics is now playing a key role in healthcare industry, pharma industry, and the pharma industry, they are constantly considering data science and bioinformatics as an important part of their portfolio. So this latest survey here shows you that AI and big data, they have like the biggest impact in pharmaceutical industries. So most times when you see pharmacists, biologists, they always have data science and mathematics skills in their portfolio. Yeah, so to really understand the application and to be able to find information from big data, it's important to learn about analysis strategy and get experience working on real data sets. So together with our research collaborators from top universities, we have prepared projects that you can go through as a project-based approach. We're going to be checking, like going through preparing a bioinformatics project where we take data sets from high impact publications and use them for practical exercises. So it's difficult to imagine exactly what the biomedical industry will look like in a few years' time, but it's very certain that there's going to be a surge in biological data. For example, the ELVA Genome Project is about to produce a whole lot of data sets. So this data is increasing from innovative research, and that we have the International Human Cell Atlas initiative also that aims to create comprehensive reference maps of all human cells. So we also have data that's been generated from wearables, implantable technologies, devices, and apps. So it is sure that we are having an explosion of data sets and this is going to like revolutionize biology and the need for data scientists in biology. So considering the exact application, I'd like to expand your mind into how computational biologists actually work out those things. How do they actually go into this analysis? How do they interpret their data? Because bioinformatics involves you getting the data, analyzing it, interpreting it to make sense to biologists and the audience at large. So, for example, this paper by Competis et al. Was using like Bayesian statistics and Markov random chain model to like understand protein-protein interaction. So we can see here, depending on distances, they're computing distances between proteins, and in a multi-dimensional scale, they're able to like see what proteins are interacting, because you really need protein-protein prediction is important in biological fields or chemistry fields. You want to know what proteins are interacting to give you that is giving a particular expression or particular function. So you can also use statistical. I don't know if we have some people that read statistics in many of our, like I've taken statistics courses. So you can apply these statistical courses, these informatics tools in understanding biological reaction like protein interaction, protein distances. So they also generated something that looks like an ontology analysis here, where using different statistical tools, they can predict what's the function of this protein. So if this statistical tool is telling you this is a DNA replication protein, how confident is it? So we have different backwards of different methods that they use to like generate protein functions, protein prediction. So that these are some of the high impact of research that people use computational biology.
I'm fine. How are you?
Okay, can you mute yourself? Okay, I think that's it. So during this program, we will explore examples from cancer research, viral infection, and microbiome composition. These are areas that have impact on all aspects of our lives. So this project will give you an insight into data set that you can start exploring to develop a research question about how these conditions are diagnosed and causes them to appear and develop, or how to treat conditions like food allergy, food intolerance, or whatnot. So let's look at big data in oncology. So big data is really helping clinicians and researchers in precision oncology. So for example, you can have a patient giving a tumor sample to a lab scientist. This lab scientist completes the genetic and protein analysis and sends the data to the bioinformatician. So the bioinformatician analyzes the sample for omics data, mutation patterns using their research tools. So they find some important genes or proteins that are significant to tumor conditions. So from this result, based on omics data that they've derived from that tumor, from that sample from the patient, a drug therapy is recommended that closely aligns with that patient's molecular profile. So that's what we call precision medicine or precision oncology, because you are taking information from this particular patient to prescribe a drug for them. It's not like a general drug prescription, but you're taking omics data from that individual to see what variation they have, what mutation they have that's making them have this kind of disease. So this will include testing FDA approved and non-approved drugs and using targeted therapeutic regimens. So the physician and the patients, they are provided with additional evidence, including adverse reactions, failure rates, and other treatment trade-offs that they should consider when making a decision in the treatment journey. So the patient and physician can in turn provide information to like strengthen the database to be used for future patients. So this is what they do in precision oncology, getting information from different patients, putting them to database, using them to get more information to for drug repurposing or drug prescription for different individuals. So some of the projects where they've obtained data for the various analyses, this publication here. So in year 2016, this research was published in OmicsLogic by Bradford et al. So Bradford et al. Explored all transcriptome profiling of patient-derived xenografts as a tool to identify both tumor and stroma-specific biomarkers. So they investigated RNA-seq as a hypothesis-free tool to generate independent tumor and stroma biomarkers. And they were able to like explore tumor-stroma interactions by exploiting human neuron compartments. So like what's the specificity of this compartment for patient-derived xenografts? So across a pan-cancer cohort of about 79 PDX models, they were able to determine that mouse stroma can be separated into distinct clusters and each corresponding to a specific stroma cell type based on omic profiles of the samples. So they didn't have an hypothesis, like a data-driven research. So they were able to get this information and separate this data into different tumor or stroma-specific cluster. So in their study, authors mainly explored breast and lung cancer datasets using PDX model. And then you can check this complete publication that is related to patient-derived xenografts if you're interested in cancer biology. So in this program, we will try to perform various NGS analysis, like the sequencing of data from next generation sequencing machines. And then we use using data sets from PDX project. So what exactly is a PDX model? So PDX models are generated when fresh tumor tissue are obtained directly from patients, and then they are implanted subcutaneously into immune deficient mice. So as such, these models maintain the principal histological, clinical, and molecular characteristics of the original patient's tumor. We are not doing it in vitro where there's probably changes to the tumor cell, but we are inserting it into mice. So you maintain the histological properties of the data and it remains biologically stable. So since PDX model closely resemble and encapsulate tumor growth in humans than standard in vitro cell line, they remain very key for preclinical drug development. So we'll try to analyze breast cancer subtypes using PDX model data. So breast cancer subtypes include the ER positive or the triple negative. The ER positive, estrogen receptor positive breast cancer is identified by the presence of an estrogen receptor on the cell membrane. Then for the triple negative breast cancer, they have none of these common receptors. So these like the basic difference between them, and then you can analyze this data, get this information, and identify this is an ER cell or a triple negative cell. And they have different levels of virulence. So one is more aggressive and one is less aggressive. So this is the information, this is the kind of information that bioinformatics analysis can give you. So we'll take data set from publication modeling precision treatment of breast cancer. And this paper was featured in Genome Biology in 2013. So this is an interesting paper to me because the authors assembled the data set of about 50 different breast cancer cells. And this represents the whole spectrum of breast cancer variations. We have human genetic variation, right? So different cancer types vary from each other. So they were able to like get 50 a representation of all the different cancer types that we have. So those cell lines have genomic, transcriptomic, and proteomic data, as well as an efficacy measurement of commonly used and experimental cancer drugs. So they're able to get those different data at different omics levels. So there's something called multi-omics data analysis. You can get different omics levels for a particular data sample and be able to analyze and get good information from it. So this is cancer study is quite interesting when it comes to like bioinformatics and computational analysis. It has helped in drug development for cancer patients. So at this point, we would like to take questions again and see what questions again. What there are.